Hi, thank you for joining me today. My name is Jamie York, and I'm an Applications Chemist at Restek in the LC department. Today, I'll be talking about approaching cannabis potency testing method development with a main focus on LC-UV instrumentation. First, some presentation objectives before we get started. First, I'll give an overview over the exciting new developments in the legalization for cannabis in the United States. And then we'll move on to cannabis flower samples and how we can prepare them and do the subsequent potency testing. And then we'll talk a little bit about concentrates, THC isomers, and how we can streamline potency testing. And then I'll give a general overview of sample preparation techniques. And then we'll talk about some samples that rely heavily on sample preparation, which are CBD-infused lotion, balms, and creams, and the full method development for this and the subsequent potency testing. Before we jump into the analysis of these different cannabis samples, I want to touch on some exciting developments in the legalization of cannabis in the United States. So as of 2020, 5 additional states have been added to the list of recreational legalization. These states include New Jersey, Vermont, Arizona, South Dakota, and Montana. So the more states that legalize the use of cannabis really allows for more opportunities for entrepreneurs to invest, more testing labs are going to open, and there's really going to be more acceptance in the industry. Hopefully all of this is leading to a path forward for testing standardization and really a very exciting future for the industry. Now let's jump into the analytical portion of this presentation. Cannabis flower samples are obviously the bread and butter of the industry. It's extremely important to be able to quantitate cannabinoid contents in these samples with the utmost confidence. So really, this is a routine, easy, straightforward analysis that potency testing labs do every day. Kind of everybody has their own flavor or way to go about doing it. So for our analysis, we use 200 milligrams of ground flour. This could be scaled up, and that really depends on the requirements for your state. So this is measured out into a 50 milliliter centrifuge tube. 10 milliliters of isopropyl alcohol is added. Again, there's other extraction solvents that can be used for this. I've seen, a mixture of methanol water, ethanol. There's really a lot of different ways to go about preparing these samples. So then the sample is vortexed for 5 seconds and sonicated for 5 minutes. This is then repeated 2 additional times for a total of 3 cycles. The sample is then centrifuged for 5 minutes. An aliquot of the supernatant is taken and diluted tenfold with solvent. And then the sample is vortexed and transferred to a filter vial and then injected onto the instrument. The subsequent analysis of these samples is very straightforward. The top chromatogram is representative of a cannabis flower sample that was prepared by the previous method, and the bottom chromatogram is representative of a 16 cannabinoid standard analyzed in solvent, just to confirm the retention times of the real sample's peaks. Both methods were analyzed on a Raptor ARC18 150 by 4.6 millimeter column using an isocratic method. The isocratic method conditions are 25% water containing 5 millimolar ammonium formate, 0.1% formic acid, and 75% acetonitrile with 0.1% formic acid. The detector used for this is a UV vis at 228 nanometers, and the overall analysis time is 9 minutes. But what about new cannabinoids? There's over 150 phytocannabinoids that have been discovered thus far. 2 of the most recent discoveries include THCP and CBDP, which were discovered last year. The difference between their structures and their parent structures is the number of carbons contained on the side chain. So here I have an example of a THC structure up top, and so you can see this has 5 carbons on that side chain. And the bottom structure is THCP, and this has 7 carbons. So this actually means that THCP has a stronger affinity for the CB1 receptor. This actually results in THCP being 33 times stronger than THC. So you can see how this is a really exciting discovery for the industry. But how can we go about detecting these compounds? we were able to develop 3 different methods monitoring 21 cannabinoids, including both CBDP and THCP, to meet various labs' needs. All 3 of these methods use the same mobile phase conditions outlined in the previous method, using an isocratic conditions and a UV-Vis detector. So all 3 methods use the Raptor ARC18 phase, which is robust and perfect for high throughput applications. The ARC18 is not a standard C18, it's a sterically hindered C18 and offers similar but some unique selectivity. So the top method uses a 150 by 4.6 millimeter column dimension and a 1.5 mil per minute flow rate. The overall runtime is 12 minutes, and this is a great, rugged, reliable choice. The column dimension actually makes it so it's transferable to older instrumentations that may not be optimized to control extra column volumes. The middle chromatogram uses a 150 by 2.1 millimeter column dimension and a 0.4 mil per minute flow rate. The overall runtime for this method is 10 minutes, cutting down on the overall analysis time from the previous method. It also uses significantly less solvent and less sample, and we refer to this method as our solvent-saving method. So the bottom chromatogram uses a 100 by 3 millimeter column dimension and a 1.8 micron particle with a flow rate of 1 mil per minute. The overall analysis time for this is 5 minutes, making it the most rapid of the 3 methods. So some considerations need to be taken into your system's extra column volume before choosing the middle or bottom column dimensions. Now let's move on to another sample type, concentrates. Concentrates come in many different forms, consistencies, and compositions. Depending on how they're prepared, it kind of depends on what will be found in the final product. So it could be prepared dry, water-based, solvent-based, or CO2. So the 2 most common classes that are found in concentrates are cannabinoids, which are potency tested by LC, and terpenes, which are typically monitored by GC analysis. So since this is mainly an LCUV presentation, we're just going to stick to talking about the cannabinoids of these. So for sample preparation, it's pretty straightforward. 20 milligrams of concentrate is measured out into a 50 milliliter centrifuge tube. 10 milliliters of ethanol is added. The sample is then vortexed for 5 seconds and sonicated for 5 minutes. This cycle is repeated 2 additional times for a total of 3 cycles. Then the sample is centrifuged and an aliquot of the supernatant is taken and diluted 50-fold with solvent. And then the sample is vortexed and filtered using a filter vial. Since we're discussing concentrate samples, let's touch a bit on THC isomers. There's a total of 30 different isomeric forms of THC that have been discovered thus far. I've listed a few up here of current interest. Of course, everyone knows delta-9 THC. It's the main psychoactive component found in cannabis. Delta-8 THC is another analog and is also psychoactive, but it's potentially less potent. The initial passing of the Farm Bill in the United States didn't initially include language for Delta-8 THC. It allowed it to kind of fly under the radar, but it's since become illegal to convert CBD to Delta-8 THC in several states. XO-THC is typically not found naturally in cannabis, but it could potentially be an indication of synthetic THC or the byproduct of manufacturing processes of concentrate preparations. Delta-10 THC is becoming a hot topic and is thought to not have psychoactive effects, but testing is somewhat limited and it could have diluted psychoactive effects. So here, a method has been developed to baseline separate these 4 THC isomers of interest in just under 4 minutes. So this method uses a Raptor fluorophenyl 100 by 3 millimeter column. The flow rate for this is 0.8 milliliters per minute with an isocratic mobile phase conditions consisting of 5 millimolar ammonium formate plus 0.1% formic acid in water, and for our organic mobile phase, 0.1% formic acid in methanol. So by using this method to look at THC concentrates, labs can cut back on instrument time due to the rapid analysis, but also save money by using methanol as opposed to acetonitrile. Some cannabis matrices rely on sample preparation more so than others. There's so many different things that cannabis has been added to that are now available on the market. Name a personal care product or type of edible, and I bet you can find it infused with cannabinoids. This makes it difficult for analytical chemists to come up with methods for each type of matrix, but it's important to remember all the tools available in your analytical toolbox. Some of the available options for cleaning up a matrix include things like solid phase extraction or SPE, centrifuging, filtration, liquid-liquid extraction, catchers, dispersive solid phase extraction or DSPE, and precipitation. If you're wanting to look at low levels of compounds, maybe something like pesticides or mycotoxins in cannabis flower, you could opt for a pre-concentration step like SPE to reach your desired limits of detection. And then another tool that you could choose, maybe if you found a wonderful extraction solvent for your analytes of interest, but it's not compatible with your LC mobile phases, you can always dry down the sample and reconstitute it with a more compatible solvent. One set of samples that relies heavily on sample preparation are CBD-infused lotion, balms, and creams. These samples contain many different components. I've listed a few of the most abundant ingredients up here for each sample type. But what really makes these challenging is that even, lotion to lotion sample or balm to balm sample, the ingredients can vary wildly. And so it really makes it challenging to come up with a method to be inclusive for all of these types of matrices. So just to name a few of the top ingredients of some of the samples we looked at, for lotions, they had things like aloe vera, water, shea butter, fatty acids, plant extracts, fragrances, and many, many more. Balms are going to be contain beeswax. That's kind of what gives it that waxy consistency. Of course, they have many other ingredients in there, fatty acids, esters of fatty acids, triglycerides. A lot of them have menthol. Have cream ingredients, so these are going to have less water than lotions and typically be thicker. They'll have emulsifiers, glycerides, fatty acids, among other things. So when developing a method for these types of samples, it's important to find a balance between the solubility of the matrix and the extractability of your analytes of interest. So now we're going to do a deep dive into the method development for these sample sets. So the overall goal of these experiments was first to find a universal extraction solvent for all lotion, balms, and creams that are infused with CBD. So we want to simplify the sample preparation and make sure any steps that we put into the sample preparation procedure have purpose and we're not doing them for too long or too short. Then we want to develop a method to accurately quantitate CBD in these samples. And then lastly, we want to make sure that we're producing a clean extract and that this extract won't dirty the instrument or the column over time. So a jumping off point for these samples, we have a starting sample preparation procedure and some starting instrument conditions. So to start off with the sample prep, a half of a gram of sample was added to a 50 milliliter centrifuge tube, and 5 milliliters of methyl tert butyl ether was added. And then 50 microliters of the sample, after it was vortexed, 50 microliters of the sample was transferred to a centrifuge tube and the solvent was dried off. And then the sample was reconstituted with solvent and vortexed and then centrifuged for 30 minutes at 4 degrees Celsius. This was transferred to an LC vial and injected onto the instrument. So for the instrument conditions, again, we're using a Raptor ARC 18 column, the 150 by 4.6 millimeter dimension. We're using a guard column cartridge for this just to help protect our analytical column, ensure that we are getting the full use out of this column's lifetime. The overall flow rate of this is 1.5 milliliters per minute. And we're using the same isocratic mobile phase conditions as previously outlined. So pictured here, we have our lotion, balm, or cream sample dissolved in our extraction solvent, our methyl tert butyl ether. You can see that it's not completely soluble. Just about all the samples had some type of precipitation or flocculation in it. The percent recovery for these experiments ranged from 75% to 95%. So there is some room for improvement here. So listed here, we have our samples in the left-hand column, our experimentally calculated milligrams of CBD per bottle, and then the standard deviation for these experiments, the label value, and our calculated percent difference from the label value. So highlighted in red here, I just have anything over 20%. We want to stay between 0 to 20%, 4% difference if we can. So we have two of these samples, one lotion and one cream, that is above our target point. Some initial solvent scouting for this. So we wanted to find a universal solvent that's going to dissolve all of these different samples as much as possible. So if they can't completely dissolve the sample, we want as small as particles as possible just to increase that surface area. So a number of different solvents were tested. These include methanol, toluene, hexane, ethyl acetate, IPA, MTBE, acetone, acetonitrile, and isooctane. Co-solvents were also tested. This is just qualitatively seeing what will dissolve best. Ultimately, we came up with a mixture of MTBE, IPA, ACN, and hexane to dissolve the samples as much as possible. So down at the bottom in the pictures, we have the MTBE samples labeled in red, and then labeled in the white numbers is going to be our mixture of co-solvents. So you can see for the most part, these samples are dissolved pretty well, a lot better than with MTBE alone. Now samples 4, 5, and 6 are going to be our bomb samples, so that's likely going to be beeswax down at the bottom. Now let's start with sample preparation optimization. So now we're going to start off by see if we can optimize the centrifuging and winterization step. So previously we were doing this for 30 minutes. So we want to test the percent recoveries in our new solvent by looking at them at 5 minutes, 10 minutes, 15, and 30 of centrifuging and winterization, and then seeing if we could maybe just do away with that stuff altogether and just use a filter vial with no centrifuging and no winterization. Here we have our results from those experiments, our percent recovery on that y-axis. So we're looking for a range from 110 to 90 to 110. On that x-axis, we have each of our 7 samples that were tested. And then the key over here in the lighter blue color is going to be our original method. So our MTBE solvent, winterizing and centrifuging for 30 minutes. And then below that is going to be our mixture of solvents with our various amounts of time or completely doing away with the centrifuging and winterization altogether. So looking at these results, overall the mixture with the filter vial had the best results. If you look in between the 90 to 110% lines here, only really lotion number 1 was a little bit out of range. Now let's move on to the quantitation of CBD. So just to kind of throw a little bit of a monkey wrench into this, we wanted to add some additional CBD containing samples just to see how well our method worked. So we're adding 4 additional samples, 2 lotion samples, 1 cream, and 1 balm, with varying concentrations of infused CBD and wildly different ingredients. So let's see how our method that has been previously developed holds up. So our sample prep right now is 1/2 gram of sample with 5 milliliters of our mixture of co-solvents. We vortex that and then centrifuge for 5 minutes. Take an aliquot of the supernatant and transfer that to a vial and dry down the solvent. Then we reconstitute that sample with 1 milliliter of solvent and use the filter vial before injecting onto the instrument. So these are our results for our developed method with our co-solvents for extraction. On the left-hand side, this is going to be our 11 different samples that we have. Beside that is going to be the experimentally calculated milligrams of CBD per bottle, and then followed by the standard deviation and our label value. And then finally in the right-hand column is going to be our percent difference from the label value. So anything highlighted in red is anything over 20%. So if you look at this data, you can start to notice something interesting. So out of the 5 samples that have high percent difference from the label value, 4 of these are actually bomb samples. Since bombs had a large percent difference from the label value, we wanted to go back and reevaluate our extraction solvent. So during the preliminary solvent scouting, we actually observed that toluene dissolved the bomb samples really well. And so pictured below is each of the samples dissolved in toluene, and 4, 5, 6, and 11 are going to be bomb samples. So here highlighted in the blue box are going to be our results from using toluene as a solvent. And in this pinky purple box are going to be our previous results with our co-solvent mixture. So looking at this data, when we look at the percent difference value, it actually got a lot worse, even compared to our previous analysis using our co-solvents. So now it's time to take kind of a new approach to this. So instead of thinking about what's going to completely dissolve these samples the best, let's find something that CBD will actually extract into really well. So we know CBD is very soluble in methanol. So let's try using methanol as an extraction solvent, even though in our preliminary test we know that it's not going to completely dissolve the samples and we are going to get some flocculation. So now using methanol as our extraction solvent, we can run through the process, basically weighing out our half gram of lotion, balm, or cream, adding 5 milliliters of methanol. I actually found a paper in a literature search that used 95.5 methanol water with 0.1% formic acid, and they seem to have really good results with this, so I decided to try that. And so after adding the solvent, the sample is vortexed and centrifuged, and then an aliquot is transferred to a vial and dried down. A 1 milliliter of solvent is added and vortexed, and then it is transferred to a filter vial before being injected onto the instrument. Here we have our experimentally determined concentration of samples for the methanol solvent. We have our experimentally calculated CBD per bottle, our standard deviation, and our percent difference for the label value. So we can see this is an improvement on some of our previous solvents. We still have a few of our samples that are higher than our 20% cutoff. So here I've gathered all of the data for the extraction solvents that we've used thus far. So if you remember back for the MTBE data, we only had samples 1 through 6 when we were doing the MTBE solvent. So we don't have data for samples 8 through 11. But all the rest of the data is compiled here. So on the y-axis, we have percent difference, and on the x-axis is each number sample. So when we look at this, we can start to see that, obviously toluene was the worst extraction solvent that we tried. The majority of these looked really, really bad. But whenever you start to look at MTBE and methanol together, you can kind of get to see some really good data going on here. So for sample number 1, MTBE looked good. For sample number 2, methanol looked good. For sample number 3, methanol. Sample 4, MTBE. 5 really could go either way. 6, MTBE looks a little bit better. And then we don't have that data for the rest of these, but methanol looked pretty good for the majority of these except for sample number 10. So between those two solvents, most of the samples could give values within a 20% difference of that label value. So we want to go through the sample preparation procedure again and perform it with MTBE just so we have data for all of the samples, but also perform it with MTBE methanol in a 50-50 mixture for the extraction solvent, as this could very possibly be our universal solvent that we're looking for. So these are our results for our mixture of solvents, our methanol MTBE at a 50-50 mixture. So looking at this data, again, we have the sample name, our experimentally calculated CBD per bottle, standard deviation, and percent difference of label value. So lotion number 1 and lotion number 10 are pretty high, but the rest of this data is starting to look pretty good. Unfortunately, at this point, sample number 6 was consumed and we couldn't replenish that sample, so we just kind of had to drop that from the analysis. So putting all this data together, again, looking at these percent difference graphs for each solvent tested for each sample. Again, we have our percent difference on our y-axis and our sample number on the x-axis. MTBE data was replaced with the most recent of data whenever we went back and redid all of that. So you can see that the methanol MTBE at a 50-50 mixture, the green bars, are looking pretty good for the majority of these samples. Really only sample 10 and 1 need a little bit of work. So let's go back and optimize our sample preparation procedure and see if we can work with sample number 1 and sample number 10 and see if we can get these samples looking good by optimizing our procedure. So using a half gram of lotion, balm, or cream again, adding our co-solvent, methyl terbutyl ether methanol mixture, and then vortexing, we're going to do a step in there where we try and optimize time on shaker table. So after we add everything together, we're going to do no time on shaker table, 5, 10, or 15 minutes. And then we'll centrifuge it for 5 minutes, take an aliquot of the supernatant and dry off the solvent, reconstitute it with a different solvent, and then we're going to put that centrifuging back in and see if that will start to help with these two samples. So we'll test this at 0, 5, and 10 minutes before transferring an aliquot into a filter vial and injecting that onto the instrument. So these are the results for our previously outlined experiments. So up top is the bar graph for lotion number 1, and at the bottom, the blue one is the bar graph for lotion number 10. On the y-axis is calculated milligrams of CBD per bottle, and that green dotted line is going to be the actually reported value, so the value that's on the bottle. On our x-axis, in the smaller subgroup, we have no centrifuging, 5 minutes of centrifuging, and 10 minutes. And then in the larger sub portion of the x-axis, we have time on shaker table. So no shaker table, 5 minutes, 10 minutes, and 15. So when looking at this data, you can start to see some really interesting trends going on here. So basically, time on shaker table had the most effect, with no time on shaker table having some of the data that was largest away from the actually reported values. So overall, centrifuging didn't have much effect between the subsets, whether you did no centrifuging, 5 minutes, or 10 minutes. So ultimately, 5 minutes on the shaker table and no centrifuging was added to our sample preparation procedure. So our final sample preparation procedure for these CBD infused lotion, balms, and creams was half a gram of sample with 5 milliliters of our methyl terbutyl ether and methanol 50-50 mixture added. Sample is vortexed until dissolved as much as possible. It will not dissolve all the way for some of these samples. And then the samples are put on a shaker table for 5 minutes and centrifuged. And then an aliquot is transferred to a vial and the solvent dried off. Then the sample is reconstituted with solvent and filtered with a filter vial. So this used a number of sample preparation techniques, including using an extraction solvent, centrifuging, drying off the solvent and reconstituting with a different one, and filtration. So the analysis of these samples is pretty straightforward. Raptor RK-18 150 by 4.6 millimeter column is used with an isocratic method and an overall 5-minute analysis time. The mobile phases include the same isocratic mobile phases as previously outlined for a cannabis flower. It's important to always use a guard cartridge when analyzing real samples to help protect your analytical column and improve the lifetime. A representative chromatogram can be seen for Lotion at the top, a balm sample in the middle, and a cream sample at the bottom. So the CBD peak is eluting a little bit after 3 minutes, and the overall analysis time for these is 5 minutes, just to ensure that everything is eluting off the column from the matrix and there's no excessive buildup. So here we have our results from our final sample preparation method. So our percent recovery is ranging from 90 to 108%, and our percent difference from our reported value ranges from 0 to 20. So in summary, it can be beneficial when potency testing to have tailored methods for each type of matrix. It can cost time in the beginning to develop these methods, but by using tailored methods for each type of matrix, labs can really save instrument time and ensure that the best quality data is being obtained. It's also important to remember that different labs have different needs, and while some labs may be most interested in saving money on mobile phase usage, others may be most interested in shortest methods and highest throughput of samples. And then finally, sample preparation is an important step in the method development process, and it's really essential for complex matrices, as we saw for our Lotion, balm, and cream samples. This is going to be the key to keeping instruments up and running and obtaining reliable and reproducible data, as well as maximizing the lifetime of the analytical column. So I'd like to thank you for your time today and for joining me, and I'd like to open it up to any questions from the audience. If you think of a question later, feel free to send me an email. I've listed my email here on the slide.